Hi, prime audience today. Have a good speaker. How are you? Good. So you know about the Blitz presentation? On
Yes.
Thursday? Yes, okay. Are you ready to do that?
No.
Okay,
See, no, I had to work overtime this weekend,
oh,
so I was really busy.
yeah, busy is fine, it is possible, so we can talk about things. So
Yeah.
would you rather
You
maybe,
are here.
I'm here, would you rather do it later or
Yeah.
or not at all?
I mean,
What's your feeling?
I'm not the most excited about presentations, but if you feel like it would be a good thing, I'll do it. But I feel like next Tuesday might be enough time for me to just get it together.
Yeah, I'm happy to give you extra time.
Okay.
So it's part of the credit.
Yeah.
So if you want to get across for credit, you have to do it.
Then I'll
If you're uncomfortable, that's fine.
Yeah.
You should learn. But
Yeah.
Okay.
Thank you.
Thanks, Lara. Did you set her up and everything?
Yes, I'm all set.
You're all set. You have a microphone?
Yeah. First one?
But since I'm here, I can make an introduction.
Thank you. Nice talk, by the way. Thank you. I enjoyed the talk. Yeah.
So good morning, everybody, also online. Don't know if you can hear me. I think this microphone should work. Yeah. So today we have a guest lecturer, which is absolutely exciting. So Natalia Komarova from UC San Diego, she was planned to give 2 online talks, but she's already here in person. So then we thought we make her first talk or her first presentation today, where she can be in person, at least with us here in the room, and still online for the people who are online. And she will talk to us about stem cells after she finished her muffin. So I need to talk a little bit more so she can get her muffin.
Introduction. Thank you, thank you. I'm happy to be here, always happy to teach, because that's what we do. So today, oh, you have heard my talk, so you don't have to listen. But good, good. So by the way, yes, please ask me any questions at any time. I really appreciate it, and I'm always happy to answer. So I will talk about stem cells, and the goal, the eventual goal, of course, is to understand mathematical oncology, and stem cells play a very important role in cancer. But we will start. So it says today's topics, but I'm going to go in more detail than my usual talk. So we will go through several of these today, and the rest of them the next time. I will start by introducing the concept of adult stem cells, and then explain something that we created over many years, which is what we call near equilibrium calculus of stem cells. It's a new mathematical tool that allows you to model stem cells and understand their dynamics and incorporate biological evidence in a systematic way to study the behavior of stem cells. And this is all about healthy stem cells. I'll give you a couple of examples, biological examples, and I think we won't go beyond this today. So let's begin by talking about stem cells. Everybody has heard about stem cells, but usually in a public discussion, we talk about embryonic stem cells. So this is things like cord blood and things like this. This is something that babies have, that embryos have, the fetus has. Very primordial, multipotent cells that divide and create all of the human body eventually by differentiation. So this is not what we'll be talking about today. It's very separate and important topic. Today we talk about adult stem cells, and everybody has them. Right? So these are organ specific, unlike the embryonic stem cells that are kind of multipotent. These stem cells belong to their organ. They do not perform the physiological function of the organ, so they're useless in terms of, say, protecting you from the sun or digesting your food. They don't do that job. They are themselves protected in a biological entity called niches. They are immortal, and I'll talk about that. And what they do is they serve as a reservoir of cells to replace and replenish the used cells of each organ. So here is my first example. I wonder if I can use a pointer. No, probably not. Like this, like in a mouse. So maybe I can bring it here. Sorry, if you bear with me a minute, I forgot to plug it in, and I'm afraid that it will run out of battery. Just 1 second. So what you see on the screen is a picture of an epithelial organ. Epithelial tissues are very important in cancer. We will talk a lot about them. One example is skin, and this is what you see on the screen. Another one is colon. We'll talk about that too. These are cells that are the most susceptible to cancer because they undergo a rapid turnover. They divide and they die, and then divide and they die. And you know that for cancer, this is kind of a breeding ground, right? Cell divisions lead to mutations, and that may result in cancer. So epithelial tissues, or tissues that divide and die all the time, are the most dangerous places in the human body for cancer. So this example of the epithelial tissue, which is skin, illustrates the structure of all such tissues. And you can see that the layer of skin shown here consists of cells of different types. They all look very different. So here, so this is the surface of the skin. The sun shines from here. And these are the cells that actually protect. So when you go to the beach and you get a little sunburned, these are the cells that are removed. You shed them off. They come off. If the sun goes a little bit deeper, you may affect these cells, and then you can have redness and some burning, okay? But the stem cells are here at the bottom, and they never see the sunlight. They are protected. They sit there in little niches because they're arguably the most important cells in this hierarchy. So in what sense is this a hierarchy? You can see this axis here. It goes from young to old. So a stem cell can divide and create a cell of a different type, right? And these cells from, this is the Latin name for this compartment of cells, kind of the deep cells. They divide and then can create offspring that kind of goes here. These cells divide and produce offspring that goes to the next layer. And finally, they create this last layer, right? This is the protective layer of the skin. And you can see that this is trying to do the justice to the physiological and biological shape of these cells. Okay, so you can see that it's a hierarchy and it goes from young to old. These cells have undergone many divisions, and eventually they will leave even without you getting sunburn. Okay, so if you get sunburn, they leave quicker. If you don't get sunburned, they will leave eventually. We shed skin all the time. That's why we have dust in our houses and apartments. This is a lot of it is these cells that are just removed. So this organ undergoes a constant turnover, which consists of hierarchical divisions of cells. And these are the cells in the last hierarchy. So so to summarize, stem cells give rise to more differentiated cells. So the process of creating different cell types is called differentiation. And stem cells are there forever, right, until we die. The differentiated cells will die eventually, right? So that's why we say the stem cells are immortal. They don't have this fate that awaits them. So all these cells will die. And this is normal, it's good. It's called apoptosis. It's a programmed cell death. They commit suicide, but it's a good thing. They do it for the sake of the organism. They've done their function, they've fulfilled it, they go away. Stem cells don't do this, so they're immortal. Okay. Now, adult stem cells and their lineages play an important role in cancer. We'll talk more about that. And let me now go over other examples of lineages that exist in human body. So I showed you skin or epidermis. This one is hematopoietic, and it's one of the most elaborate hierarchical trees in the human body. Hematopoietic means blood. So what do we have here? We have the stem cell that is here at the top of this hierarchy. This way, for this picture, I reversed it. So the stem cell is on top of this hierarchy. So it divides, and actually this diagram skips several steps because it's even more complex than this. But there are two lineages that kind of result from the stem cell. This is called myeloid stem cell, and this is lymphoid stem cell. This cell gives rise to all these different types of cells that are found in the blood. And you will be familiar with some of these names because they come up in a standard blood test. We'll go to the doctor, take a blood test. They'll count the platelets. The red blood cells here, and some of these neutrophils, they measure, right? So this is something that you can measure in the blood, and they all have different shapes. If you look in the microscope, you can recognize them by their different shapes. So this is this lineage that makes up your blood. And this other lineage makes up your immune system. Also the cells that will be found in blood. And you can see that these are T cells, these are B cells. If you ever studied viruses and the immune system, very familiar names. So yeah, two branches of this lineage. And an interesting part is that the stem cell is not even in the blood. This stem cell is sitting in the bone marrow, in a niche. It's protected. These cells die all the time, and they're being replenished by divisions of these blood-producing stem cells that reside in the bone marrow. So these cells are the ones that we say perform their functions. They carry oxygen, they protect us from viruses, being part of the immune system. They carry nutrients and all that. And this is the stem cell doing nothing but replenishing this army of workers, right? And there are interactions between them. And the focus of this work is to study these interactions. Okay, so of course, question.
Both the myeloid and the lymphoid stem cells live in the bone marrow.
So
Or is it
these
like-
these are still in the bone marrow, and these are in the blood. So these are in the bone marrow. These are in the bone marrow. These are in the blood. Very, very good question. We will focus on the maintenance of nearly constant cell numbers in the body. So in the zoo, right, the number of these cells has to be very steady. You know that from your blood test. If the number is up or down, bad news. So the goal of the organism to maintain a constant size. And that means health. In biology, maintaining anything at a constant is called homeostasis. And it's not necessarily like cell numbers. Temperature that is constant is homeostasis. Anything that's constant. So in mathematics, we say equilibrium. In biology, they say homeostasis. So here, these lineages, one of their tasks is to maintain homeostasis, to be as constant and as steady as possible. And the boss of that, the cell that's responsible for this is the stem cell. And that's what happens when the organ is healthy. Now a mutant can come up that refuses to respond correctly to these signals, and it refuses to keep homeostasis. And that's bad news, because this may be the first step toward cancer. So the goal of such systems is to cooperate with each other, to act in sync, and to maintain a steady state. And that means for these guys, killing themselves. So from the point of view of a selfish cell, that's not a good idea, right? If I want to be selfish, I don't want to die. I want to keep dividing. Evolution is based on that, right? So multicellular organisms have evolved to kind of the cells learned to ignore their own good for the sake of the collective, for the sake of the organ. These cells have to die. They have to be replaced. If they refuse to die, that's bad news. If this thing starts dividing out of turn and making too many, that's even worse. Okay, so that's cancer. So this may be the first step toward cancer. So let me show you some other lineages that are there and are characterized by stem cells. So this is colon. As I said, this is another example of an epithelial tissue. So imagine colon as a tube. Let's look at the wall of that tube. And the wall of the tube looks like this. It consists of folds of skin or epithelial tissue that are organized like this. This is a cross section. These are three-dimensional folds. If you look at them in this direction, they're circular like this. So they're kind of protrusions. Right? So this is a cross section of them in a longitudinal direction. The number of cells in one of these, so these are called crypts, colony crypts. The total number of cells is about 10 to the 4, about 10,000 cells. And so here is a magnification of this, and they can be divided into compartments in a similar fashion as we looked at before, but it's simpler. So here at the bottom, there are stem cells, and actually they talk to these other cells here that don't belong, technically speaking, to the crypt tissue, but they're nearby, right? So they're not there in isolation, but they have this nice little niche where they reside. They proliferate, creating this next layer and the next layer and the next layer. This process takes about a week. After a week, these guys, they sit at the top of the crypt. They are the ones that interact with the food. They digest your food. After a week, they die. So this whole process takes about a week. And there are many, many more of these compared to those. There are about like 5 of these, like very small number. But that is 5 per crypt. The number of crypts in the human colon is 10 to the 7. So that's 10 million. So we have 10 million of these little folds. Each of them has several stem cells. Okay. And you see no hierarchy here, no branching hierarchy like in the blood. So it's this kind of more like a linear progression of cell types. And these are called transit amplifying cells. And these are called fully differentiated cells. So TA, transit amplifying. Okay, neural. So these are stem cells that are responsible for the neural system. And you can see that this lineage, although not as long as the hematopoietic lineage, also branches into 2. And we have neurons here and astrocytes and oligodendrocytes. Okay, different types. And the shape, it's supposed to, do justice to the shape. So you can see that even though all these cells have exactly the same genome, there is no difference. They look completely different and they do different things. So it's not about the genome, it's about how it deals with this genome. It's the expression of the genes. So something changes when we go from here to here. The expression of the genes changes. They make different proteins. They become, they do different things, and they have different shape. Olfactory. So olfactory means sense of smell. So these are located in the nose, and it's a linear progression like this. And they create neurons that are responsible for detecting smell and processing the smell. Okay. So, and the question, so I mentioned that the main thing kind of what is being done here is maintaining a constant size. You can ask me what can be easier, right? Maybe it's very difficult to change size, but maintaining a constant size seems like an easy task. But in fact, it's not. So, and I'm going to explain why it may be a difficult task. I'm not very, yeah, okay. So let's look at the simplified picture where we only keep track of two types of cells, stem cells and differentiated cells. You saw that it's not true for any of the examples that I gave you. There are at least like 3 or 4 different types, but let's just consider stem cells and the next one up, right? Okay, so this is your organ, and let's suppose that one of those differentiated cells either died or like divided and went to the next compartment. Okay, so there's a space left, okay, because it needed to divide. It needed, otherwise nothing happens, right? So there's a space left, and we remember that it's the stem cell's duty to fill that space, to replenish, okay? So what is the stem cell going to do? There are 3 things that it can do. And this is the first place where mathematics comes in. Right? So there are 3 types of divisions that have been described that stem cells can do. The first, let's start with this one, actually. That's the simplest. This is proliferation. A stem cell divides, and both daughter cells look exactly like the mother cell. So there are now more stem cells. There was one, now there are 2. The second process here at the bottom is what they call differentiation. A stem cell divides, but something miraculous happens, and both daughter cells look different from their parent. They are more differentiated. So here they are denoted by the color red. So as the cell divides, something kicks in biological, and the gene expression in the daughter cells will now be different from that of the mother cell. And this type of division is the weirdest, right? So this first one that I described is like what all cells do, right? They just divide and they increase the numbers. This division is coupled with differentiation. And this is an asymmetric division. A stem cell divides, and one of the daughter cells is like the mother, and the other one is differentiated. And this is unique to stem cells. This was first described in the fly Drosophila, and people used to think that this is what stem cells are all about. But it's not necessarily true. In fact, all 3 types of cell divisions occur in humans and in animals. And sometimes it's even more often than not that something like this happens. But now, okay, so the question mark here stands for which division the stem cell will undertake. Let's consider all 3 possibilities. So if it's this division, let's track the number of stem cells, so the dark blue cells here. If there was X of them, after this division, X goes to X, the number of stem cells does not change because this mother cell will give rise to itself and a red cell. So the number of stem cells does not change. If this type of division is called proliferation division. And you can see that in this case, the number of stem cells actually increases by 1. Well, there was 1 here, now there are 2, and we'll have a blue cell here. And in the case that we have differentiation, this blue cell will be replaced by 2 red cells. So there'll be a red cell here and a red cell here, and the number of stem cells will go down. Okay, so let's suppose that all the processes are happening in the system. And let's suppose that there is a certain probability that proliferations happen, and there is a certain probability that differentiations happen. So if the probability to decrease the number of cells is greater than probability to increase, think about it. I'm more likely to decrease the number of cells than increase. Then eventually, the number of stem cells will go down, and this organ will have no stem cells. That's death, okay? Not a good outcome. If we have the reversed inequality, and the probability to increase the number of stem cells is larger than the probability to decrease, then the number of stem cells will steadily increase because they're more likely to go up than they are to go down. and the number of stem cells will expand. And of course, that's bad, because any expansion in an organ of an adult is bad, right? So not a good idea. So the only choice is to have them equal to each other. So I'm as likely to differentiate as I am to proliferate. So this last line corresponds to these two processes exactly balancing each other. Okay, so that seems like a good idea. But now, I don't know how many of you have studied stochastic processes. This here is exactly a symmetric random walk, or what they call a gambler's ruin problem, or a drunkard's walk. So I can explain it to you very simply. So let's suppose that I'm standing here and I can take steps to the left and to the right. And let's suppose the probability to go right is the same as probability to go left. So I'm going to engage in a motion that is random, and I go right and left, and sometimes by chance I go twice right, and sometimes like this. So I'm going to start kind of randomly going up and down. That's why it's called the drunkard's walk, right? Because the drunk person, you know. Okay, so, and one of the properties of this type of motion, you go up and down, up and down, up and down, but statistically speaking, you're going to go off to infinity. It's just a fact. As time goes to infinity, this process will take you far away. It's not as efficient as a direct walk, where I always take steps in the same direction. But it gets you there. It gets you away to infinity. So in this most, reasonable process that we assumed, when the probability to increase of the number of cells is the same as the probability to decrease, if they're equal to each other, then you are going to, the number of cells will engage in a drunkard's walk, and either they will disappear, like in this implementation of this process, or they will blow up, right? So both results will happen with equal probability, and it's not good. So something is missing from this model, but it illustrates why it's difficult to maintain homeostasis. We now tried three logical possibilities for these probabilities, and all of them led to bad outcomes. So something else is needed, right? And what is needed is a feedback. There must be something in the system that tells me if the number of stem cells, for example, goes too low, there must be some signal sent that says, oh, come on, start dividing. There are too few of us. If this number of stem cells is too large, there must be another signal that says, oh, come on, stop doing this. Otherwise, there are too many of you. So there must be a signal, and there must be a response, right? The necessity for these signals have long been recognized in biology. So I'm going to go through several examples of signaling that has been assumed or studied in different systems. So these are excerpts from various papers that look at various stages of the stem cell hierarchy, and they map out signals that are sent or received by different cells. Okay, so this one is a theoretical paper that assumes, these arrows here, right, the blunt arrows, they, so let's, for example, look at one of them. So we have the probability of self-renewal, right? This is the probability when a stem cell divides and creates two of the same kind. So this round arrow is the probability of that decision. And you can see that it's negatively regulated by stem cells, transit amplifying cells, and differentiated cells. So 0 means stem cell. This is the next member of the hierarchy, and this is the last member of this simple hierarchy. The number of these cells negatively affects the probability for the stem cell to make two of itself. And so in this model, it was assumed that this rate is negatively regulated by everybody, and this rate is negatively regulated. What does it mean negatively regulated? It means the more, say, of these cells they are, the less this process will go on. And I'll show you later. So this is an assumption of this model, okay? That's another model that appeared somewhere else, and they assume these other types of control, right? It gets pretty complicated. This is a simple one, feedback inhibition. This is another one. So many papers assume a certain number and direction of these arrows that show the direction of the signal. Who sends the signal, who receives the signal, and what process this signal affects, right? So there's more, and everybody assumes something else. The problem is that it's not really known, right? It's very difficult for biologists to figure out exactly what happens in the cell's mind. It doesn't have a mind, but it has to make a decision. What do I do now? What happens inside the cell which makes it either divide symmetrically into two differentiated cells, or self-renew, or go an asymmetric route? What happens? Nobody knows. So what we want to do here is kind of create a theoretical framework for all such processes, right? And then we see that some of them are better than others. So this whole branch, we call it near equilibrium calculus of stem cells. So it's clear why it's about stem cells, because that's what it's about. Near equilibrium means that we are not talking about a sick person that has cancer. It's far away from the equilibrium, something is growing. We're talking about a healthy organ that has homeostasis. So this calculus is near the equilibrium. Why is it calculus? Because it's simple, okay? So let's set it up by using the following very simple decision tree. So this is a stem cell, and remember, I'm only using this language to explain it simply, but of course, a stem cell does not have a brain. It doesn't think. It just responds to signals. But a stem cell sitting in a niche may do a couple of different things. So we call it stem cell fate, or the decision that it makes. It can either divide or not divide, right? That's a decision, a very simple one. So let's suppose it decided to divide. Yes, it divides. Now it faces the decision to either differentiate, so create two daughter cells that are both differentiated, or it can create two cells that are the same as the stem cell. So differentiate or not. Yes, we'll go here. No, we go here. A differentiated cell is shown here separately. In the simple system, it doesn't divide, and it's often the case that fully differentiated cells never divide. They just die at some point. So the only decision that they have to make is to die or not to die, right? And that's a decision. It's a programmed cell death. It wants to die. It's called apoptosis. So yes or no, it dies or not. So in this simple example, these are the only decisions that there are. And now we say, okay, there's a population of cells. So each individual cell may receive a signal that's sent by the whole population, and it will influence which way it goes, yes or no, right? For example, very simply, if I'm a stem cell and I'm thinking, should I divide or should I stay as I am? And there are many of stem cells around. In fact, let's suppose there are too many for some reason, and they all send me the signal, don't divide, because there are too many of us. So I respond to that signal and I don't divide. And that's how you control the numbers, right? And here, if I decide to divide, should I differentiate or not? If there are too many of these, they're going to send the signal, stop, don't differentiate. There are too many of us. So that's how the system can control itself. Okay, so how do we model this? Let's suppose we, again, in a very simple case, just have stem cells and one step removed differentiated cells, and we'll call them X and Y. So this cell has to decide, do I divide or not? So divisions are happened at a rate, and this rate is basically the rate at which it decides to divide, right? And once it decides to divide, it can differentiate or self-renew, okay? So when it differentiates, it makes these red cells, If it doesn't differentiate, it makes 2 of those. Right? And this guy can decide to die. So this is the same kind of picture as in the previous slide, but kind of more spread out. And now we can draw controls. So here's an example of 4 control loops. And in this case, stem cells send the signal to themselves. And they say the more of us they are, the more we should divide. So it doesn't necessarily make sense. It's just an example of control loops that we could draw, right? So in this picture, stem cells send the signal to themselves and say, the more of us they are, the more you should divide. Okay. And they also send a signal to themselves saying, if there are more of us, let's not differentiate. So that's a negative arrow. Okay. And the number of differentiated cells send the signal back to stem cells, encouraging differentiation and discouraging division. It's an example. Okay. So mathematically, we can express this as a derivative of the rate with respect to the population. So this first arrow says that the rate at which cells divide depends on x and y, and dl dx is positive. The more stem cells they are, the more they divide. That's what this arrow means. Okay, so this is positive. And how does it depend on y? Let's look. Y's prevent them from division. So the derivative with respect to y is negative. The more of these cells they are, the less stem cells will divide. So these arrows, positive, negative, translate directly into the sign of the partial derivatives. Or we can change these arrows, right? We are here to draw all the possibilities because we don't know what's out there. I just changed this. Now I have only 3 loops, and this one is negative, this one is negative, this one is positive. And the signs changed, and this is 0. Now L is not controlled by X. I removed that arrow. So, right? This is the formalism, mathematical formalism that translates me drawing these arrows into some mathematics, right? And you can see that there are many arrows you can draw, but not an infinite number of arrows. You will run out of possibilities. There could be different combinations. So the question is, which combinations work and which don't, right? So let's figure out how to study this. Here we have a system of ordered differential equations, which talks about the number of stem cells and the number of differentiated cells. And it takes into account the rates that are listed here. So this is the rate of division. So with rate L, these cells decide to divide, and with rate 1 minus p, they self-renew. So p is the probability that when they divide, they make red cells. So that's not good. So we're looking at the change of x. So if they decide to differentiate, that actually takes out cells from x and moves it to y's, but with a factor of 2. This is the rate at which stem cells decided to differentiate. So they make 2 of them, and that adds to y. With probability 1 minus p, they decided to proliferate. So that adds to the pool of x's. And finally, differentiated cells die with rate d. So that takes away. Question?
So for your L1 minus P term, this is really minus L1 minus P and then plus 2L1 minus P, right? So kind of you lose the mother and then you get 2 daughter stem cells back.
Are you talking about this term or this term?
Right.
First term?
Yeah.
So this is the rate at which stem cells divide, and they divide into their own pool, right? With probability 1 minus p. And when an X stem cell divides, it makes 2, so 1 extra. So this comes with multiplier 1. So it adds 1 cell. Each such division adds 1 cell. Was that the question?
Yes, okay.
And when it decides to differentiate, it means that this cell, it decides to become red. Okay, so one of the blue cells is taken away from here, but 2 red cells are added to the pool of differentiated cells because it divided, right? So that's how it works. And this is simple math. You can simplify this to that, right? So it goes, it's L minus LP minus LP. So it's L minus 2LP. So this is your simple equation. And L and P and D are functions of X and Y. We just don't know what functions they are because nobody knows that. Okay, so a system of two ODEs we can solve. Oh, and here it says that this is the process of proliferation, differentiation, and death. Okay, so when we have an equation like this, the first thing to do is to find equilibria. The first equation, let's solve that. We have 1 minus 2p equals 0, which means that p equals 1/2. So that's the solution of the first equation. This tells me that at the equilibrium, the probability to differentiate should be exactly half. I'm as likely to differentiate as I am to self-renew, right? So the two types of stem cell divisions have to be happening at the same rate, okay? And this here comes from solving this equation, and it says that the rate at which I decide to divide should be equal to the rate at which I decide to die. And actually, these are different populations. This is the rate of divisions of X, and this is the rate of death of Y, okay? But to maintain homeostasis, the two have to be equal. And remember again, these are functions of X and Y. Okay, so we can find the solution X, Y of those equations, and that's your steady state. And then we can figure out its stability properties. Okay, so there's also a stochastic model. Question?
Another question. So this steady state doesn't have to be unique, right? You could have like
steady state,
16 steady states or something.
When we write it like this, there's of course no indication of uniqueness or even existence of the state. If we just say, oh, these are most general functions of X and Y. But we assume that these functions are such that there is a reasonable equilibrium that does correspond to the homeostasis, right? So we have to make sure that these functions are reasonable. And in everything that follows, we assume that an equilibrium, a positive equilibrium exists, right? Otherwise, there is no organ, right? You can also approach this stochastically. So you probably know very well by now that in mathematical biology, there are different ways to model nature. So this way, ordinary differential equations is everybody's favorite because it's so simple. You can just write these ODEs, you solve them, do stability, but it has disadvantages. One of the disadvantages is that every time you solve an ODE, you get the same answer. But every time you grow bacteria in a dish, you get a different answer. So something is wrong between this, like philosophically wrong, with trying to describe a biological process with ODEs. It always gives you the same answer. So if we want to take account of the randomness of nature and how things may go different ways, we have to apply different mathematical tools. And so this here is an example of a stochastic model. And this tool is much more complex, and you need to take more classes to master that, but it gives you an advantage of having different outcomes every time you run the system. So you can do it here, right? So here, these are real decisions that a stem cell takes. If it has a 50% chance to divide, to self-renew and to differentiate, it will really do it like every time it flips a coin, right? So this process lets you flip a coin at every step, okay? And I won't go into details. Now, another thing that you can do in this approach is to make it more realistic. Before, remember, we only had X, the stem cells, and Y, the differentiated cells. What if we have 3 different types? Like in the colon, often they approximate these scripts as stem cells, transient amplifying cells, and differentiated cells. So this is what's drawn here. 3 types: stem cells, transient amplifying cells, differentiated cells. Now there are more processes, more decisions, and more arrows. So you can draw these different arrows, right, and kind of see what happens. Right.
Sorry, Natalia. One more question.
Yeah, of course.
You mentioned the stochastic modeling just 2 slides ago. Are you going to do more about this in this lecture or not? because I will touch it later in October again. So, so maybe I could just tell
should
the
we go over
students.
that slide? I'll be happy.
No, on the contrary, I wanted to tell the students that I will go over this method later in the class.
Oh, so I shouldn't.
Yeah.
Okay,
So,
sure. No, I wasn't planning on it. Very good. So is there a question? No. Okay, so this is the stochastic model. Good, fine. So here's an example of how this may work. So let's assume that we have two different compartments, stem cells and differentiated cells, and only three processes, the rate of division, the probability of differentiation, and the rate of death. This stochastic model allows you to calculate not only the steady state, how many cells you are going to have, it tells you the variance. So if you just solve an ODE, right, and you come to a steady state, you have to look at me. What happens is like your solution changes and then it stabilizes, and as a function of time, it remains constant. So this is what ODEs do if they have a steady state. There's a limit. The solution approaches to a constant. In a stochastic process and in nature, it's not like this. You're going to measure something. It's going to go up and down, up and down. And the best you hope for is a steady state that kind of oscillates around a constant value. So it's not going to be a constant. This is the mean, the average, but it goes up and down, up and down. Question is, how much does it go up and down? Does it go up and down like this? Or does it go up? Okay. Presumably, in a living organ, we don't want it to do this. We want it to do like as little as possible. So the characteristic that quantifies the amount of jumping is what they call variance. And you can calculate variance based on this method by using the controls, right? Okay. So we can calculate the moments. So the variance is the second moment. The mean is the first moment. You can calculate all the moments. So with this framework, we're going to answer different questions. For example, what's the smallest number of controls that maintains homeostasis, right? I told you that we can draw many arrows for different processes. So what's the smallest number of arrows? And the answer to this question is 2 in this one. So if we have just stem cells and differentiated cells, you don't need 4 arrows. Like there is nothing that says that the number of stem cells controls the probability of differentiation, or the number of differentiated cells controls the rate of division. Right? It's just that, or this. So, and there is nothing else. So these are the minimum control loops that are compatible with homeostasis. If you want to be economical, you don't want to spend too much of your energy signaling. So let's just step back for a second. What are these arrows? Controls. What does it mean that the population controls the process? It means that each cell in that population makes a signal. The signal kind of leaves the cell, and by the process of diffusion, it kind of spreads around. And I'm sitting here, and I can sense the signal that comes from that population. It could be my own population, it could be another population. I sense it, and I act accordingly. So in order to send the signal, you have to make it, and you have to like send it out. That's expensive. So you presumably you don't want to have too many of those. So this is the most economical way to control homeostasis. Stem cells prevent their own divisions. The more of them they are, the less them divide. And differentiated cells prevent differentiation. The more differentiated cells they are, the less is the probability of those guys to differentiate. That's type number 1. Type number 2 is completely different. Stem cells encourage differentiations. The more stem cells they are, the more they tell themselves, we need to differentiate. And that makes sense, right? I don't want to have too many of my own type. Let me differentiate. And differentiated cells stop divisions. If there are too many differentiated cells, they tell those guys, you already should stop dividing. Enough, right? So in this, there's nothing else. So these are the most economical ways to control the system. And you can prove it mathematically. And of course, we can generalize it. We are mathematicians. Let's suppose we have n different compartments. We have stem cells, and then the next, and the next, and the next, like in those complicated lineages that I showed you before. If there are n compartments, the minimum number of control loops is n. So if I have 5 different types, I have to have 5 different arrows. Each of the compartments must mediate at least one control. So like here, stem cells send a signal and differentiated cells send a signal. If I have 5 of these, there must be an arrow that comes from each of them. And the probability of differentiation must be controlled. There must be at least one arrow that controls differentiation. So, and you can prove that this is the minimum number of signals that you can send to control the loop. So going back to the question, is it difficult to maintain homeostasis? You can see that it's difficult, right? There are all these theories, completely not trivial. Like, why is that, right? So this follows from this very general mathematical approach. Now, if we have 3 types of cells, stem cells, transit amplifying cells, and differentiated cells, this is the simplest model of colonic crypts. There are 20 loops that are the minimum loops that can maintain homeostasis. So this is an analogy of this. Here I have stem cells and differentiated cells. These are the smallest loops that can control it. If I increase the number of cells to 3, I have this. And actually, this is a simplification. There is another 28, altogether is 48, that can control death. I excluded them from here. So, so you can see that there is a lot of variety of different ways to control the system. And if I increase the number of cells to 4, complexity just blows up. There are thousands of different types of control loops that could be there. And these are minimum controls. You can see that each of these only has 3 loops, 1, 2, 3. Remember, I told you, if there are n compartments, the minimum number of controls is n. So there's 1,000 other types of control, but they'll have more loops. They're like redundant controls. This is the most economical way. So the question is, which one of this occurs in nature, right? There are a lot of possibilities. Which one are we going to take? This is the work that we did with the help of data. And this is the first time I'm using data in this talk. And it shows you how this theory can be applied. So the experimental data consisted of 3 probability distributions or histograms like this that came from a biologist, David Axelrod at Rutgers, who studies colon. So these numbers are the number of stem cells, the number of transit amplifying cells, and the number of differentiated cells in 49 crypts. So they took crypts, colonic crypts from dead people, and they counted how many different types of cells were found in those. And there is a variety, right? You can see if everybody had exactly the same number of transit amplifying cells, like 600, then there would be just one column here, like a very huge one. There would be 49 people who had 600 cells. You can see that they're distributed. So some people had 600, some people have 400, some people have 200. So it's naturally variable, right? And this has to do with my depiction of a stochastic process, right? It's not always the same. And if we catch different people at different moment of time, you're going to see difference, right? Okay, so these are the data. And the question is, if we have these distributions, what control system is there that's compatible with that? Kind of a difficult question, right? Because it's not a whole lot of information that we're given. We're just given the distributions of these numbers. Okay, so it turns out that we can narrow down the possibilities just based on this information and some other relevant biological information. And I'll show you how to do that. So we start with 20 different control loops. And we want to exclude, right, based on the data, which ones should not be there. So the first thing that we excluded are non-local control loops. So what's non-local? Let's look at number 3, for example, here, this one. Here, stem cells control themselves, that's fine. And transit amplifying cells control their own probability to differentiate. But differentiated cells control differentiations of these guys. And remember, the crypt has this vertical structure, spatial structure. stem cells sitting on top, differentiated cells at the top, stem cells sitting at the bottom, and differentiated cells on top, and there's 10 to the 4 of them. Like there is a large relative distance between the stem cells and the most differentiated cells. So we assume that these cells are too far to control those. So we're going to exclude all the loops that have these non-local controls. We just say that I can either control myself or my neighbor, and my neighbor can control here or here, but we never allow these cells to control. Yeah. Okay, so these are gone. Only local controls. Okay, so what's next? So let's look at the actual data that was given to us, the means and the variance. So I told you that this theory can tell you the mean and the variance of the steady state given the controls. Now we reverse the problem. We know the mean and we know the variance from this. We know the mean and we know the variance from these distributions. Can we solve and find the controls that can do that? So when you solve this, you find that sometimes the control loops are characterized by like imaginary numbers or negative numbers of cells. And that means that there is no solution to this. It means that these means and these variances are not compatible with a certain arrangement of controls. So whenever we get this non-result, like impossibility, we cross them out. Okay, got rid of those. Question?
Can I ask you another question? So
Of course.
I have the advantage, I hear this for the second time. So you gave a similar talk last week. So I could think about this a little bit more. In your conditions, you only talk about the partial derivatives like qx
Yes, the sign,
negative,
the sign of the
dx
derivative.
is positive.
So these correspond to signs. Yeah.
Yeah, but it seems like I can choose the p whichever way I want and make the variance huge. So how do you get information about the variance if you don't know the size of p of x? It could be 10,000, or it could be 1 million.
Yes.
So I think you get a huge
Nonetheless, you
variance.
can get a restriction based on the signs of these things. So these arrows, so this means that dp dy is less than 0.
Yeah.
So just that allows you to
So it could be minus 10 million, and you still get
yes,
small
it's only
variance.
minus the minutes, but it gives you like an imaginary. I'm sorry. So you start with the given solution for the mean and the given size of the variance. So these are real numbers. You calculate them in terms of the controls, and now you solve for the controls given these values, and you get a negative, and you get an imaginary number.
Mhm.
So your partial derivative is imaginary,
Oh, I see.
and that's when you throw it out. Yeah.
Okay.
So there's mean and variance. Let's suppose there are only two, and there's like Lx, dl, dx, and dp, dy. So we can say that these are my controls. They create this, or we can take these as known and solve for those. They're the same number because they're and if this on the solution, we know these numbers for these. The structure of the equations follows from the loops, and if we get an imaginary number, we throw them out. Yeah. Okay. So we don't need to know the values of these derivatives. We solve for them. Very good. So those are thrown out. Oscillatory injury recovery. So it's known, and it has been measured in mice, that if you remove one of the cell types, it will grow back, but it will grow, not remove, like decrease, right? If you injury means that you take away some of the cells. It will grow back, go back to the equilibrium, but it will go up and down before that. Up and down more so than the usual fluctuations. So there are some oscillations in the system, and oscillations mean imaginary part of the eigenvalues of these systems. So if you ever done a stability analysis, you look at the Jacobian of these systems and you calculate the eigenvalues. They must be, they must have a non-trivial imaginary part. So that oh yeah, this is the picture actually. I forgot I had it. So these are oscillations that were measured in a real system. So you can see that it goes up and down. So we have to get rid of those that do not allow for imaginary eigenvalues. Okay, and there's the last thing is intra crypt correlations. So the data that we received can be plotted in a different way. I think I didn't do this here. What is the dependence of the number of differentiated cells on the number of transit amplifying cells? So we can present these points that I gave you not as histograms, but as correlations. And if they're positive, negative correlations, it should be taken into account. These simulations of these loops exclude some of these if they give you a wrong type of correlations. And in the end, we find ourselves having only two types of regulation left, okay, out of 20. And actually, I repeated this analysis with an extended set of 48. In the end, you also get something like this, plus one that controls the depth here. But so, and you can see that they're very similar. So in both of them, stem cells negatively control their own division. So if there are too many of them, they are going to stop dividing. Transit amplifying cells, don't let them differentiate. If there are too many of them, they say, okay, you can divide, but just keep in that compartment. Don't come here. And the fully differentiated cells, in this case, tell these guys, stop dividing. And in this case, they tell them to stop differentiating. And that's the only difference. So these are very similar loops, and they seem to be compatible with the data. So I want to advertise this method. So anybody who ever wants to do biological research, it's a very cheap method to look into nature. So the expensive method is to use biological experiments and try to measure the signals that are observed in the crypt. So you have to find those chemicals that control various processes. You have to figure out what these chemicals do. And people do this. They look at these substances, they dump them on their system, and they watch what happens, right? Oh, the divisions increase, the differentiations decrease. So, and they also have to figure out who sends the signal. That's the hardest part. Actually, we know what different chemicals do when you dump them on a system. And most experiments are like this, but we don't know where they come from, which kind of cells sends them. So this is the expensive and difficult way. This is the cheap way. Mathematics provides a very cheap way to kind of look into the functioning of systems and see what's going on there. Okay, so this made us think about principles of control networks that may be there that govern the choice that nature makes, the evolution makes in this area. Economy is what we think restricts the number of loops. So as I said, it's expensive to send these signals. So maybe we should, try to get away with as few as possible. Robustness, or I'll talk about later, and minimization of variance is what I just is the principle that allows us to kind of get rid of the wild spread in the number of various populations. So this is the next topic. And I think I have about 20 minutes. So let me just do that and we'll be done for today. This is another application which has to do with a different stem cell system. It's airway epithelium. So this is a picture from the nose, epithelium tissue in the nose of a mouse. And these cells are there to purify the air that the mouse breathes. So they're stem cells. They don't do anything. They just replenish the system. And there are two types of more differentiated cells that perform their functions. And they're different in nature. These are secretory cells. They secrete some gooey stuff such that the dust in the air gets stuck and doesn't go into the lungs. And these are ciliated cells. They have kind of like a brush or like some hairs attached to them. Those hairs also catch the dust, so that purify the air. Okay, so this is an attempt to model this system, which consists of 3 compartments again. X is stem cells, Y is secretory, sticky cells, and Z is ciliated or hairy cells. And what they tell us is that this hierarchy is not necessarily linear. So in column, we knew that these gave rise to those, and those gave rise to those, linear. And that's the simplest, actually. Here, there are a couple of more things that could happen. It's possible that these cells directly give rise to those cells. And even more weird is these cells can go back and become stem cells. So they can kind of transform their properties. And you see, it's not easy. These are these cells, and they can somehow change their status and go back. And this is a very useful mechanism, actually, to have. If some injury depletes the pool of stem cells, what are you going to do, right? What happens is these cells kind of sense this, again, through signaling, and they de-differentiate and replace them, okay, and become stem cells. But at the same time, it's very dangerous, because if the mechanism of them going back is too much, they will all turn into stem cells, and that's cancer, because these cells are immortal, right? They don't want to die. Okay, so we can imagine all these different processes that happen there, and put that in our analysis. And that includes symmetric and asymmetric divisions, differentiations, and de-differentiations. Okay, lots of processes. We were trying to figure out which ones are really there, right? And these are the experiments that we had available. There are 3 sources of experiments, all published in very high-profile journals. The first one is this. Let's remove ciliated cells here. And then nothing much happens. So that's a whole paper published about that. The system does come back, but it takes forever, right? So there's no immediate reaction. If we remove both Y and Z, both types of differentiated cells, the system recovers fast. If you remove X, the stem cells, again, the system recovers fast. And that's it. We don't know what happens if you just remove Y. There is no such experiment. So these are the 3 pieces of information. So we thought, okay, depending on these arrows and controls, like you remove one population, and so the control signal changes, and we can watch how the system reacts and what happens. It turns out that no matter how you draw these arrows, you can reproduce these results. Like you can find parameter values, which we of course don't know. You can find parameter values that have a slow recovery if Z is removed, and a fast recovery if Y and Z are both removed, and a fast recovery is if X is removed. So that really didn't restrict anything. And then, so we started imagining, okay, what kind of like minimum organization can there be? For example, only symmetric divisions. Symmetric means that there's no, you either differentiate or self-renew, no asymmetric divisions, or only asymmetric divisions, or both, but such that stem cells have only asymmetric divisions, and these other cells only have symmetric divisions. So you can check different ones, and you can calculate the variance. So again, the variance is the amount of spread. And we kind of check this and that, and we're able to show that this specific type of organization minimizes variance. In this case, the stochastic system stays at the equilibrium and oscillates the minimum around it. Okay, and then good news came. So this network minimizes the variance of stem cells. And then we found this paper that actually measured everything. And they found that it's the exact way in which this system is organized in the mouse body. It has so red means negative control, blue means positive control. So this is a positive control of this differentiation rate by axis here. This is a negative control of the death of ciliated cells by themselves, and so on, right? So we found this as a candidate which minimizes all the variances. And this is exactly what happens in the body. So that's why we now believe that this may become principle, one of the principles of how the bodies are constructed, what multicellular organisms are constructed. And maybe with further research, we should not forget that, right? We should not forget that this is probably one of the constraints on the architecture of the bodies. Okay, so the next thing that I will start now and finish next time is breaking networks. So this is about robustness, right? So it's all nice when everything is great. The healthy organ can maintain its own size and also kind of oscillates around it. But every time a cell divides, there could be mutation. A mutant cell, most of the time, does absolutely nothing. Actually, most of the time it dies. So most of the time a mutation leads to cell death. It's removed. Nobody worries about it. Sometimes it stays, but it doesn't do anything. So it's just a change, a small change in the genome, but there is no effect. And very rarely, it's a change that leads to a phenotypic change. Phenotypic means that the cell has different properties, and maybe it's a selfish cell. If it's a selfish cell, it doesn't want to cooperate with others. It may be cancerous, right? It may want to start dividing too much, or not dying, which is kind of the same effect, right? So we want to explore this by looking at different networks that exist and how a mutation like this can break it and kind of grow out of control. And the question is, what are better networks and what are worse networks from the point of view of cancer? Maybe some are easier to break, and those should be avoided at all costs because we don't want cancer. Maybe some networks are harder to break. So this was the mathematical question that we addressed in this paper. I would say it's not a final answer, but it gives you some hints at what happens. And we examined a relatively simple network of only 2 types, but we included many processes, right? So, so what processes are here? These cells can die. These are stem cells. They can self-renew, so they can divide into 2 X's. Yeah. They can differentiate by dividing into 2 Ys. They can asymmetrically divide by dividing into an X and a Y. And Y, the differentiated cells, can also self-renew here. This is an assumption that sometimes it doesn't hold. If these are terminally differentiated cells, they just do their job, they don't divide, and they just die. But here we include this process for completeness. We say that they can divide, they can die, and they can transform back to become stem cells. So I think there's nothing else that one can think of. These are all the processes that what one could list that happen to cells. Okay, so we include them all. And we write down this process, the equations for these compartments. So let me quickly go over them because I think it's kind of simple. So X is the number of stem cells. Dot means derivative in time, so the rate of change. So differentiations, that's when these cells divide and create 2 of these. That comes with a minus sign. So it leaves this compartment. This is the death of stem cells. It goes out. So I wonder if this is a mistake. I think it's the wrong sign. So this should come with a minus sign. This should come with a minus sign. So this should be plus here. And this should come with a plus sign, should be minus sign here. Oh, great. I've given this talk before, never noticed this. Now I go into details and I was like, oh my God. So this is another positive term. So this is a contribution of Ys to increase the full pool of X's. So Ys transform and become X's. So we have a positive term from Y. And this term comes from here. It comes from YB. It goes out of this equation and comes back here. So it's this process from here to here. There's a 2 here. These are the cells that come from the stem cells. They differentiate and become 2 Ys. So that's a 2. this is asymmetric division. So stem cells divide into one stem cell and one differentiated cell. So it simply adds one of the differentiated cells, and differentiated cells die. Okay, that's all there is to it. And we can combine some of these processes together because they come with the same sign. So we only have 5 rates, and these are the simplified equations. Okay, so they're combined rates. So these are the ODEs, not so bad. There are 2 ODEs, but we don't know the functional form. We always keep everything general. R is a function of x and y. S is a function of x and y. We don't know what it is, but maybe we know the sign of the derivative.
There's a question.
Oh, okay. Where is it? What? Where is the contribution of asymmetric division on the x equation? Excellent question. So let's suppose that we have a stem cell and it divides. This daughter cell is x, this daughter cell is y. So what happens to the number of x's as a result of this division? There was 1 cell and there's still 1 cell. So it does not change. This type of division is the only one that doesn't make any difference to the number of stem cells, but it adds 1 differentiated cell. Is that clear? So this is an excellent question, and you have to think about it before you write the equations. Okay, so what are the minimum loops that maintain homeostasis here? This is a different problem from the one before because we have more processes. We have back divisions, all this stuff, right, that we added. So it turns out that altogether, there are 10 different loops that can be here. So these first 4 does not contain the rate of back differentiations. So if B is 0, we only have 4 loops. And in the presence of back de-differentiation, we have 6 more. So the back de-differentiation is kind of a rare process. It's not always present in all systems. So we did it with and without it, right? So without it, things are a lot simpler actually. And these are the 4 types of control. Here, stem cells positively control their own differentiation or negatively control their self-renewal, or stem cells encourage differentiation and discourage self-renewal. So that's it. That's all there can be here. And in the presence of B, there are other more exotic control loops. So there are 10 loops that are minimal. It means that the cells spend minimum amount of effort, send only 1 signal, right? What kinds of mutations can break this system is the next question. And I wonder, it's 11:17 now, only have 3 minutes. So I don't think I should start on this. And maybe I can take questions in the next 3 minutes, and then we'll finish next time I talk.
I think that's a great idea. So first of all, thank you very much, Natalia, for this wonderful presentation.
Thank you.
Really, really interesting.